So thank you again. My name is Ravinder Pal Singh. I lead microbiome, the division at Genomics. And my colleague Nick James is joining me here today. And he's going to tell you all the exciting stuff, and I'm going to tell you more the intro. So bear with me for a few minutes while I give you some introduction, set the scene, the sort of challenges we're working on, how we're working with Unilever, etc. And then Nick will get into the details, and there's a lot of detail he'll cover. So bear with us as we get there. Thank you. As I said, we're going to cover the intro. There's always the case of who we are. Been around for 9-10 years, sit at Hingston, UK. That's the Wellcome Genome Campus, the big campus in the UK for genomics, etc. From day one, we've been an AWS partner. We build on AWS, etc. And we've really optimized around AWS and other cloud type providers. And as you expand past human genomics and you get into things like microbiomics and metagenomics, you get to life sciences, a lot going on there. And in fact, today, a lot of what we're going to talk about in the workflow and the workflow manager are very applicable in those metagenomics, microbiomics worlds. There is a challenge. The challenge is a very real challenge. Scientists have access to a lot of data. And this data is put, just so much of it, prevalent, internal, external, collaborative, et cetera, reuse. You can't name it. There's just too much data available. Problem is scientists can't use the data. And there's a need to be able to bring that data into an ingestible form, understand the value of data and all that good stuff, and curate the data, et cetera. And these are real upstream challenges before you even get to things like analysis and big AI and things like that. To do that, you've got a lot of variability in all these different data sets. It's very complicated what's going on, and it leads to some modeling challenges and uncertainty and risks in your models when you do your analyses. These problems need to be resolved. Scientists need to be able to self-service to this, these upstream problems without the need for massive amounts of bioinformaticians, et cetera, in the system. There are certain characteristics or attributes at the bottom here I list here. You need trustworthy and validated evidence. Questions, you need to understand the questions being asked. You contextualize the questions being asked. If I'm doing a study on topic A, it's very different to doing a study on topic B. And they may involve similar types of data sets, for example, from immuno-oncology to looking at certain types of skin microbiome. They may involve reuse of certain types of data, but the nature of that use, the context of that use is very different, so you need to work that. And then data becomes an asset, and for all those folks out there who worry about buying data, generating data, licensing data, et cetera, there's a lot of OpEx being rolled out there, and you need to manage that. And then the last point here is that these are complex questions being asked in studies. So scientists are faced with having to look at the combination of data sets, not just single data sets. To roll this up into a world of what this means in terms of trends, we've all seen this, we all know this. We've gone from sitting at our desktop, in the sequencing world with a handful of sequences, a handful of sequences, 100K, all the way to billions of sequences, and there are billions of sequences being rolled out, and especially, again, if you're in the microbiomics, metagenomics, it's far more complex than human genomics, clinical genomics. It's very hard to industrialize and standardize that world, and there is just so much horsepower you need to be able to at speed, and you need things like AWS, and you need things like on-demand. And so, Eagle, as I say, has been working on this for 10 years, et cetera. And so you'll see here these terms here, e-cataloging and eHive. There's some technologies, Nick will talk about that in a minute. To split this into two sides of the coin, you've got the side which says, how do I make this seamless? How do I make this scalable? How do I put it on cloud and all that good stuff? That's the left-hand side you're seeing here. That's where we should be, absolutely need to be, and if we're not there already, if it's not industrialized for the scientists to directly use point and click, then we're probably behind the times. The thing is that with the advent of some of these AI techniques and with the statistical power we have, we can now move to a world where it's questions-based, where it's actually far more evolved, where scientists can actually present the questions they're asking, and the data can be self-service to them. And this change, this paradigm change in how data is accessed, used, and then analyzed, et cetera, is an inflection point for the way that R&D is being done in industry today. So you watch that space, please. So what we're going to do today is we're going to talk about technology which is embedded, workflow manager technology which is embedded in both these left and right sides, and the importance and how we've got to developing that technology. To put that in another context, if we're scientists today, we'll sit down and go through some analysis. It will be a fairly kludgy manual process that will meet constraints of time and cost, but innovation will be suppressed because they won't be using the data sets available to them. We understand that that's a problem. You can offer terabytes of data to a scientist. They'll only use the data they understand at hand, typically their data. So if you want to ungunge this, you have to provide a self-service approach, and hence the right-hand side of the slide I mentioned earlier. And you have to allow the system to use appropriate deep learning techniques, use appropriate on-spot cloud analyses. So you've got the OpEx working, you've got the AI working, and you're able to provide, in this case, the biologist data at hand, which they need in a very seamless, iterative fashion without armies of bioinformaticians needed in support. You build this all into an architecture. Now we're going to get to how Nick's deep dive is going to find its way in the slot into this. You've got a whole series of activities needed. You need to think about catalogs, you need to think about curation, you need to think about valuation discovery engines, you need to think about data access layers, NFRs, non-functional requirements pieces, et cetera, et cetera. There's a high-level functional architecture for you. And you can see where the red box is painted. This is where the workflow manager kicks in. And actually, we're starting to deploy the workflow manager across this flow. So the importance of speed, cost, on demand, et cetera, become extremely relative to relevant across a functional architecture, not just in certain parts of it. And Nick will touch upon that. Just mentioning Unilever's, the context of the case study. So as you can imagine, these large CPG or FMCG companies have R&D. They're doing a lot of microbiology, a lot of microbiome, lots of, whether it's 16S to Metagenomics, et cetera. And as they go through this, they're doing a lot of study work, at-scale studies, and they're trying to process this data with OpEx in mind, with scaled use in mind for scientists. So here, what you have here is a very simple view with bioinformaticians in the middle. We need to get the bioinformaticians. We need to industrialize that portion of the flow so that scientists can work with the lead bioinformaticians as necessary, but really it's the scientists running the workflows. And so to put this in a series of requirements, we have the background, the requirements. So Unilever have certain needs from their engagements at an industrial level. And as you can see here, this requires new biology to be applied, new informatics programs, et cetera, and access to public data. You need this underlying architecture must be evolvable. I mean, these are things we understand. AWS developers understand these things, but you're seeing the whole CPG, FMCG, biopharma markets waking up to these requirements. And then, of course, you need to be able to use the robust solutions that AWS offer here in this case. And you can see the outcome on the right-hand side. There's a scale at which scientists can now use the data, use the workflows, and this is what you want. You want many scientists directly tapping, directly running. You want things running much faster. You want 20 times speed, at the minimum, and you want OpEx to accordingly come down, and Nick will talk about some of that. And then, of course, you want to know that the public cloud works in these environments. You have confidential data, and these very confidential workflows. And my last slide is just to reflect that this does lead to really credible, powerful outcomes. This is a Nature Scientific Reports paper from this year, and it shows a first-of-a-kind oral microbiome a product claim. And these are very hard product claims to get, very thorough studies. Please do let us know. We'll send you the paper. It's not a problem. Contact us afterwards. And again, Pete Keeley here just commenting on what it's done for the team there. So now I'm going to hand over to Nick, and Nick will give you more details about that workflow manager. Thanks, Nick.
Raminder. So as Raminder's been introducing, I'm going to talk about how we run workflows at Genomics and how we've used the Amazon Cloud to do that. So before I go into that technical side, I just wanted to take a quick step back and think about the process that Raminder's been describing a little more. So we mentioned there's this bioinformatician. So there's an end user scientist who's producing this raw data and hand this to the bioinformatician. So what's happening at this point? So they're basically running a series of steps. And these are bioinformatics tools. There's a huge variety of bioinformatics tools. They're basically complex algorithms and a very wide range of things that they're doing here. And each one requires a high-level expertise. We need to understand the function that this tool is doing, and therefore we need to understand the parameters that are going to get the best result with using this tool. And each one can be complicated to install. They can be produced by a variety of people and so there may be varying levels of documentation and things like this. So the net result here is that this process is time-consuming and challenging for this bioinformatician. And so we sort of painted this picture of a fairly ad hoc process using lots of manual expertise and time. But actually, if we break this process down, what's really happening is we're going from step 1, and step 1 is flowing to step 2 is flowing to step 3 and to step 4. So we can define these steps, and we can document them and really understand explicitly what's happening. And once we've done that, then we can take another step and we can represent this programmatically. So we have a workflow configuration file of some sort. And of course, when we've got this far, then we've taken a very big step towards enabling reproducibility. So we're not relying on this manual sequence of steps. We can run this programmatic steps again and again. So how do we run these steps? Well, we use a workflow engine. There's a very wide variety of workflow engines out there, and they all have different advantages and disadvantages or strengths and weaknesses. So for us, in this scenario, then we have a number of very key requirements which are essential for us. And the first of these is that the workflow engine is very scalable. We're working with very large data sets, and we need to be able to scale up to hundreds of machines and cope with large data files. And we need something which is reproducible. This is essential in a scientific environment. If we take the same workflow, the same data, we end up with the same results. We need something which is traceable. We need to be able to monitor the workflow run, and we need to be able to see what each job is doing, and this is useful for development as well, of course. And we need something which is robust, because sometimes jobs are going to fail. They might just need rerunning. We need to track all this process and not just fall over at the first hurdle. And we need something which is capable of handling complicated workflows. So, for example, if we have a sequence file and we measure the quality of that in one step, then we might want to flow to a different branch if that quality is not very high, rather than flowing down the main branch and waste compute resources. So we need to handle conditional data flow and this type of thing. So at Eagle, we've based our workflow approach on an open source software called eHive. And eHive has been developed since 2004 at the European Bioinformatics Institute, or EBI. And EBI is a world-leading research institute producing and hosting a very wide variety of biology-related databases, which are illustrated in the diagram here. And eHive is involved in many of these processes in processing this data. So basically, eHive is a very tried and tested workflow engine in a genomics large-scale environment. So before I talk about how we've utilized eHive to run our workflows in a production environment in the cloud, then I just need to explain some key concepts so that we can understand the process that we needed to take. So firstly, eHive is a Blackboard system. And what this means is that when we run a workflow, this workflow template is loaded into this Blackboard database. And this database contains now the structure of the workflow and understands the flow from step to step and the parameters that are needed. And secondly, it has a record of all the jobs at the different steps. So how does eHive actually run jobs if it's not this central Blackboard process which is running them? Well, they're processed by autonomous agents, which we call workers. And in this diagram here on the right, then we have a worker for analysis A. This is a round worker, which is matching the diagram on the blackboard on the left, and we can see there's one job ready for that analysis. We also have an analysis B, which is a diamond-shaped worker, and there's no jobs for that analysis. And what happens is that the worker queries the Blackboard to see if there's any jobs that match its analysis type. And in this case, we have a job which analysis A, the worker can process. So it claims that job and starts running it, but it also updates the status of the Blackboard. And then as it's processed that job and it's complete, it updates the status to done, and we create 5 more jobs in this example. So we can see job creation is a dynamic process in eHive. So hopefully it's this is clear that this approach, we don't have a load on the central system. It's the workers which are claiming jobs and creating new jobs. So it's a very scalable design, sort of inherent to the design of eHive. So I mentioned a lot about this eHive worker. So what is an eHive worker? So firstly, it contains the bioinformatics tool. This is obviously the tool we're interested in, doing the interesting algorithm. Then secondly, we have something that we call a runnable. And basically, this is a fairly simple wrapper around the bioinformatics tool, which handles some very useful functions. And firstly, it will handle the input parameters. And so, for example, does the parameter have a value when it should when it's an essential parameter? And is that value in the correct format that we're expecting? And then it will handle the input, fetching of the input data and make that available for the bioinformatics tool to run from whatever location that's been in. And then, of course, it will run the bioinformatics tool. And again, this can be a fairly complicated process, and it's good to manage that within the runnable. So, for example, if a file is of a different type, then we might want to use a different set of parameters, and we can make that decision as the runnable is running the bioinformatics tool. And then once the tool is completed, it's created our output files, we write those to our output location. And within all of these processes we described, then eHive is logging information. So if something goes wrong in the input fetching stage, the file doesn't exist or something like this, it's logged, and we can see what's happening. So we've introduced the eHive worker. So how does eHive actually run workers? So it uses a batch scheduler to do this. So eHive monitors the Blackboard and sees whether there are jobs that are ready to be done. In this case, there's 5 ready here. And then it checks the batch scheduler, sees if there's workers already in the queue or whether there's workers already running. And it makes the decision that, in this case, we need to start more workers. So it places workers in the queue. So we're on to analysis B here, which is a diamond-shaped one. So we've requested some diamond-shaped workers in the queue. So it's a scheduler which finds the available resources. That's what it's designed to do. And when it finds a resource, then it launches those workers. So note that eHive is not running each job one by one. It's not managing millions of jobs. What we're doing is we're requesting workers to start, and they will handle the fetching the jobs. So it's quite a scalable design. So when the worker starts, then it queries the Blackboard, and they get the jobs. So we've got 3 jobs running and 2 jobs ready now. And then as those jobs completed, they're going to create more jobs. So we're on to analysis C now. And what we find is that the diamond-shaped workers, we had 3 of them, so there's only 2 jobs left. So two workers have claimed 2 more jobs, and we've got 1 worker with no jobs left. So that worker just exits. And eHive is then monitoring the Blackboard and sees there's now 24 jobs ready in the third analysis, analysis C, and this is our rectangular-shaped analysis. And so it submits these requests into the queue, into the batch scheduler, and the batch scheduler will then find resources available and run those workers. And what we can see here is that those workers will claim jobs, and we actually have jobs from multiple analyses running at the same time. So eHive is quite dynamic and perfectly happy to run different steps of the workflow all at the same time. And there's still a lot of jobs there ready to be run, so eHive will put more workers into the queue, and as the resources are available in the scheduler, then it will run the jobs. So that was a very quick introduction to some key concepts about eHive. So now I'd like to talk about how we've adapted this, and we've managed to run our pipelines in the AWS cloud, and in fact how we brought it back again. So we started with this open source software, eHive, which was developed at the UBI. And in fact, as it was developed at the UBI, they had a cluster management system called LSF in mind, which is what they have on site there. So it's primarily designed to run on LSF. And we want to make this a part of our platform. And to do this, we need to be able to run in a cloud environment. This is our approach that we wanted to take. So secondly, the original eHive, it requires a lot of technical knowledge to run. It's primarily a command line run tool. And we want to enable the scientist, who's not a software programmer, to be able to run the pipeline without any knowledge of eHive. And lastly, we want to then integrate it with other components of the Eagle Genomics platform. So going back to 2008, which is a very long time ago in the AWS world, then we adopted this approach of representing an eHive worker as an AWS instance. So it's sort of fairly straightforward. We have an AMI, which contains all the installation, and we have an EC2 instance which is running with the Blackboard on. And as it's processing jobs, it starts the appropriate AMI, an instance based on the appropriate AMI, and that is now our worker, and it grabs the jobs and processes them as I've described, and we fetch and write inputs from the Amazon S3. So I should say that this was this very early days of Amazon Cloud, and we were sort of quite early in running workflows in production in the cloud. So it was an effective approach. But there were issues with the resource usage. So we're having to specify each workflow step needs different resources, and they vary quite widely. And so we have a bit of a bottleneck where we've been using one instance type, and then we need to switch to another. We need to wait for the instance to terminate and start again. And so we can improve that slightly by using sort of an average instance type, but again, that's not ideal. And also, we have a tendency to write the whole workflow onto a single AMI, and this has several disadvantages. That it slows the development time. It's difficult to test an individual analysis, so it becomes merged into a workflow as a whole, and that means that we're not getting nice reusable analysis steps, which is what we really want. But one thing that we learned during this process, where we could be installing 30, 40 different bioinformatics tools in one workflow, is we started automating installation of the bioinformatics tools rather than doing this by hand. We used a configuration management tool called Chef to do this, and this meant that we could have a cookbook, which would be a GitHub repository, and it would contain a version of each cookbook, and we could also test that installation. So we improved the whole installation process and automated it, which was a big help. So then, of course, along came Docker, and we started to think, well, can we represent our eHive worker as a Docker container? And there are two main challenges here. Can we make an eHive worker into a Docker image, firstly? And then once we've done this, how can we manage scheduling this container across the cluster? And are there any tools that will help us with this? And this is back in 2015. So I'd like to talk now about that first challenge, making that Docker image. So we start with a Docker container here, and as I mentioned, we already had a good library of bioinformatics tool cookbooks, and so it was surprisingly easy to point Chef at building our Docker container. So we already got our installation, and we can just run that on the container. So we've got our bioinformatics tool installed, and we've also got a versioned installation for that. And using Chef, it's very easy to incorporate our runnable code, which is now a separate GitHub repository. And again, this is a versioned software. And we can integrate all these things together and the other eHive dependencies which are needed for the runnable to work. And now we can run the test that each runnable has, such as running the bioinformatics tool. So we now have the tool installed. We can actually run the test in a meaningful way with some test data. And we can also test various aspects of the runnable, such as validating input parameters and whether it's creating the expected number of jobs given the test data that we've given it. And once we've built this container, we can then mount the runnable code into the container, so we can develop that code on our laptop with our IDE of choice, and we can actually run the tests in effectively a production environment. And having got this far, then it's a sort of fairly logical step just to have a nice continuous integration environment. So when we make a commit to our runnable repository, or in fact on a weekly basis as well, then Travis will trigger a build, and Chef will build this container with the runnable and the bioinformatics tool, and then it will run the tests that we've been looking at, and if those tests pass, then it will push that image to the repository for that analysis step in Docker Hub. So just to just pause slightly and consider some questions that I found very relevant about how we're building Docker images. So firstly, do we know how the image that we're using was built? So in other words, if we don't, should we be trusting that image? And really, we should have some kind of documentation about this. And so secondly, does that build still work now? So if the last time it was built was a year ago, can we rely on that actually working now? I mean, very often the dependencies and things are going to change and the build just won't work anymore. So again, we need a process of continuous integration to ensure that we know that it's still working. And in fact, when the build works, that's no guarantee that the runnable and the bioinformatics tool is actually working. So we should be running tests on the software having built it as well. And again, continuous integration is a good solution to that. And then lastly, do we have any need to run any other container types other than Docker? So for example, if we're running in an HPC environment, then Docker's we might not be allowed to run Docker, basically. So do we need to bear in mind that as we're choosing how to build our images? So now we've got to the point that we have a workflow configuration file. It's in its own GitHub repository, and it's versioned. So we've got version 1 in this example. And then each analysis step within the workflow refers to a specific Docker image. So in this example, analysis A is pointing towards the analysis A Docker repository, and it's using the version 1 tag. So we can have multiple tags, obviously, for each Docker repository. And then analysis B is pointing to the version 2 tag in the Docker image B, and analysis C is pointing to the version 1 tag of the repository for the analysis C image. And what that means is that once we deployed our version 1 of the workflow, we can very happily go on to version 2. So we can deploy version 2 for another customer and not worry about breaking version 1, or we can have multiple versions, whatever. So in this case, version 2 and analysis A is using version 3 now. It's using a different tag. Analysis B is using version 2. So we can define all these things, and that's versioned in our workflow configuration file. So I talked a lot about versioning, and why is that? Well, it's essential in scientific workflows. Then reproducibility is key, and in fact, it's quite a significant problem now that there's sort of a number of publications where other groups are not able to replicate the work in that publication, and that's a major issue. And reasons for this are that the steps are not well enough defined, so we need to be very explicit about exactly what each workflow step is doing. And we need very clear instructions about that, and the versioning is obviously a key part of that. And this is also important in quality control and compliance and other processes. And when our end-user scientist runs a workflow, then we generate a workflow run report. And that contains a description of a workflow that they can understand. So it's not the programmatic configuration file. It's a description, diagram, etc. And it also shows each tool and version that was using that workflow. And this is really important because it might be in 3 months' time that they want to run the same workflow again with the same data, and they would expect the same results. So it's important to know which tools and versions, and they really do want to know that. So another aspect that we've added to the eHive worker is the logging and having hundreds of workers all running jobs. We need to keep track of that. So we added the ability to, using FluentD and Elasticsearch, to log these jobs to the central logging system. And once we've got the data in Elasticsearch, then obviously we can use Kibana to visualize that. It's a very dynamic way to create nice interfaces to Elasticsearch. So in this example, then we've got, in the top left, we've got the job count over time. So it's a good way of just monitoring what's going on, and then errors and warnings, and even job analysis per analysis. So now I'd like to talk about the second problem that we had, which is how do we deal with scheduling these containers. So just to briefly sort of recap, then eHive was originally designed to schedule jobs on an LSF cluster. And to do this, it uses an LSF interface. And in fact, it also has another interface. It can schedule jobs on an SGE cluster. So the obvious thing for us is that we'd make another interface, a Docker interface, and that would schedule jobs to some kind of Docker scheduler as well. So we could write this interface quite easily, but what about the clusterer? What tools are there that are going to be suitable for eHive? So this goes back to 2015, and obviously this is quite a fast-moving field, and things are different now. But at the time, we were looking for some key criteria, and it has to be a good scheduler, obviously. It needs to be able to schedule containers, thousands of containers. And yes, there were various tools that could do that. And we need something with a queryable interface. So eHive needs to find out how many workers are running. Again, that's possible. We need to be able to allocate memory resources and CPU usage before the eHive, before the worker starts. And this is really important with bioinformatics tools because they can quite happily carry on, in some cases, for an hour or two using very little memory, for instance, and then that memory can really expand and use, say, 50 gigabytes. So we don't want our decision-making to cram lots of containers all onto one machine just because they don't happen to be using much resource at the time, and then for all of them to start needing lots of memory. So we know this happens in advance, so we want to be able to allocate it. And then we need to be able to this to be able to auto-scale. So this is really important. As I've been describing, eHive can create these jobs dynamically, and at the start of a workflow, we typically just have one job. So we don't want 100 instances running to process our one job. And we need to scale down again. And there weren't really any solutions at the time that would do the memory allocation and auto-scale. We also need a queue. So obviously, if we don't have enough instances running, we don't want to lose that request for a container, so we want to put it in a queue. And again, there wasn't really anything at the time. And lastly, we needed something which is not tied to a particular cloud platform. So we have customers which have their preference of provider, so we need to be fairly cross-platform in our choice. And there were a number of tools here. So as you can see, there wasn't anything at the time that exactly met our requirements. So we decided to put our own solution together, and we called this a Docker Swarm Orchestrator. And we tried to choose the biggest sort of starting components that we can, rather than writing lots of stuff that's already there. So we looked at container scheduler, and we chose a Docker Swarm for this. This is a legacy version of Docker Swarm. And then to enable an auto-scaling feature, we chose the Docker Machine API. So this Docker Machine handles starting instances and adding them into a swarm and and terminating instances and removing them, obviously. And it's also very cross-platform. So all that cross-platform work is handled for us in Docker machine. And we needed to write the REST interfacing and queue, and basically we just did that from scratch, really. We used RabbitMQ for the queue. And so these components add up, and that's basically what makes the Docker Swarm Orchestrator. So just to show how this works in practice, then we make a container post request. This goes to the REST interface of the DSO. And then when we make the request, that DSO puts that in the queue. So now that we have a container in the queue, we don't have any instances running, so we create a new swarm. So the Docker Swarm uses a discovery service. So we use console here, and we register this new swarm. And then we use the Docker Machine API to add instances to this swarm when we have things in the queue. And then that instance is registered in Docker Swarm, and Docker Swarm pulls that image from Docker Hub and runs the container and does the scheduling bit with the resources available. So we can post any number of jobs into this queue. So here we've got some large containers with higher memory requirements and some small containers. These go into the queue, and the DSO starts the containers using resources as available. So we have more jobs, so it started more instances. So we can see we can have 4 small containers on one instance here, and just the one large container fits on and takes up a whole instance. So obviously the resource use is a lot more efficient already. And we still got 2 large containers as well in the queue. So at this point, if we query the container count endpoint, then we can see that there are 2 queue jobs and 5 running. We obviously get a lot more information than that, but this is just an example. And as we've got those 2 containers in the queue still, then the DSO starts up some more instances. So you can see how it's scaling up based on the demand here. And there's containers start running. And now in our endpoint, we have 7 running containers. So now I'd just like to put these 2 stages together and how this all works in practice. So we have a workflow with different steps, and each step needs different resources. So analysis A is 2 gigabytes, analysis B is 7.5 gigabytes, analysis C is 100 gigabytes. So now eHive looks at the blackboard and sees there's jobs to be run. This is for analysis A, so it's 2 gigabytes. So it makes a request to the DSO, and this 2 gigabyte container goes into the queue. And as we described, the DSO creates a swarm, starts an instance, and then Docker Swarm pulls this image from Docker Hub, and there's our 2 gigabyte container running. So the instance that it's pulled here is the versioned instance for image for analysis A. So it's specific for this analysis. And now this is an eHive worker, and that worker queries the blackboard and grabs that job. So we now have 1 job running on that instance. And as that job runs, it fetches the data from the eCatalog, which is stored in S3. And as it's processing, then it will write the output back to the catalog. And then we created 5 more jobs, and these will go into the queue and get processed in the same way. So these are analysis B. There are 7.5 gigabyte memory container. So we need to start up some more instances as we have more containers. And then these start up, and it grabs these jobs and processes them. So that's how we've put these components together. So just a quick note on spot instances. A great idea. We get 80% reduction on the on-demand price, so it's just a huge cost saving. And just a couple of notes that the instance type reduction can vary widely, so it's not necessarily what you'd expect. A more expensive on-demand price can be cheaper as a spot instance And also the availability zone can vary a lot. So some availability zones, such as in red here, obviously have some very high peaks, whereas others are very stable. So just need to bear this in mind. So exposences are great, we get a big reduction, but obviously they come with a caveat that the instance can be terminated at any time. So how do we cope with this in an SLA sort of production environment? Well, so with the system I described, then we have multi-layered mitigation for this. So eHive is monitoring each of these jobs, and if a job fails, it already knows how to handle this. And in fact, it's also monitoring the worker. So if a worker disappears, it's also tracking this, and it can request more workers if they disappear. So we got the DSO also has its own queue, and these jobs are not going to disappear if we lose all our spot instances. And finally, Docker Swarm works by having Docker Swarm Manager components, and if we lose those, then we could, in theory, not be able to use our swarm. So again, the DSO can handle this. It can just start new swarm managers, and we can carry on. So that's kind of an overview of the system that we implemented. So now I'd just like to talk about how we put this into practice with some real data. Here we have a tool that we were looking at called NeedData, and what we noticed as we were developing our Metagenomics workflows that this tool was taking a lot longer than we expected. It wasn't the sort of the key part of the workflow. It's kind of a preliminary step to increase the quality of the data first. Looking at these charts, what we tried is that we noticed with 7.5 gigabytes, although the tool was working, it was using a lot of memory and taking much longer. So we tried some different parameters. Basically, we gave it more memory. We tried some different parameters to the tool itself. And what eHive does is it records the metrics of each job, so we can easily see the average time per job. And looking through this table, then by changing these parameters, we've gone from nearly 7 hours to just under 1 hour. Which is a big time saving, and that's sort of interesting. But how does that translate to cost? So that's about $2.50 for this step for one sample to less than a third of $1. So that doesn't sound very much, but a typical metagenomics run could be, say, 1,000 samples. So just for this single step in the pipeline, then the cost could be $2,500 down to $300. And as I mentioned, we're using spot instances. So actually, the resulting reduction comes down to $40. So I mentioned we've written this Metagenomics workflow. And so the aim of this workflow is basically two things. It's to characterize the gene function of every sequence. And it's also, secondly, to identify the species, the taxonomy of all the bacteria based on the sequence that we have in the sample. So we're not sequencing a single species here. We're sequencing all the DNA in a sample containing many different bacteria. And the input to this pipeline was about, in this example, was about 2 billion read pairs. And it took about 30 hours across 50 instances, and that's about 200 CPUs. And total cost for this pipeline run was less than $50. So just some conclusions and perspective. So what we've enabled here is a scientist who's not a computer programmer to upload their data and curate it in the eCatalog. And in fact, they can share that data with other users and collaborate, but they or the other users can also run that pipeline from the eCatalog interface without knowing anything about eHive. And in fact, they can monitor the pipeline, and they can see all the pipeline steps and how many jobs are running and how many jobs are done as the pipeline is running. And then actually the people uploading the data can be in one country, and the people processing the data can be in another country. So all this has been a great improvement in the use of the technology, but it's been very successful in the Amazon cloud. But can we run this now in an HPC environment, for example, on an SGE cluster? So the first thing to note is the SGE cluster already has its own scheduler, and it's very happy with that, and probably having Docker Swarm as well it would be a very bad idea, as you don't really want two schedulers. So could we use our SGE interface to schedule our Docker containers across that environment? Well, many HPC admins are not very happy to run Docker in this environment for a variety of reasons. But we can use another container type called Singularity, which is very well fitted to HPC. In fact, it's become very, very popular over the last year or so in this environment. So what we have is an SGE Singularity interface for eHive, and that can actually use our Docker images and translate them into Singularity and run across the SGE cluster. So basically, we have just one workflow template, the same file, and we can run that on the Amazon cloud or on an internal SGE cluster. So we have our catalog, which can have the data in different places for different studies, and we have eHive, which is able to run the same analysis in different locations. So what this means is we can move our analytics to the data and not the data to the analytics. And this is a concept we can has been known as federated analytics and results integration. And in fact, we can actually go a step further here, and we can run, say, the first step of our pipeline on our internal compute, and then our third step on AWS cloud. So I've mentioned that we've enabled our end-user scientists to run a pipeline without having to use eHive in a hands-on way. We've got this web interface. So how did we do that? Because eHive was a fairly command-line-driven tool. So we wrote a REST interface for it, and with a pipeline, a catalog to keep track of all the runs. And this REST interface will allow the catalog to query the run status and things like this. So this is an example of how we've integrated two different components of the Eagle platform. And as Raminder introduced, this is our functional architecture diagram. So what I just showed is represented here in the workflow manager section diagram. And now we're looking at how we can integrate, say, eHive with eDiscover. And what this would mean is that we run a heavy-duty workflow analysis, and then when we get the results from that, we've got new outputs and data, and we can integrate that and link it to the sort of knowledge graph in eDiscover, and sort of weave all this data together so we can do these sort of complex queries. So that's kind of where we are at the moment now. So finally, Unilever have been using this platform, running these workflows, curating them properly in the first place in the catalog, and running them with this interface, and just the time saving and the scale that's enabled is increased by 20 times for them. So thank you very much, and please, if you have any questions.